Hello everyone. In this video, I will be speaking about how drought influences Pseudomonas syringae infection in Arabidopsis thaliana. Infection cycle of Pseudomonas syringae. Pseudomonas syringae increases its number in the presence of high water levels, which indicates that it is a water-loving bacteria. Upon infection, it senses the plant water level and damages the plant leaves. Here, in this diagram, which explains the infection process. Firstly, bacteria present on the leaf surface of healthy plants enter the leaf through the stomata, then colonize in the apoplast region and multiply and causes chlorosis of plant leaves. Influence of drought on Pseudomonas syringae. Drought reduce pathogen number in combined stressed plants show some unique and tailored responses under combined stress which was not seen under individual stress. Drought changes plant pathogen interaction by changing some biochemical and physiological mechanism of host plants and makes the plants either less or more susceptible. In this diagram, you can see drought inhibits the infection of pathogens and makes plants less susceptible to pathogen infection. Combination of drought and pathogens infection have two outcomes. First outcome is that two stresses can act additively and amplify the plant destruction. For example, drought has been shown to intensify many fungal, bacteria and viral infections in plants. And second outcome is that both stresses can act as antagonists to each other. That means presence of one stressor reduces the effect of other stressor and result in enhance the plant tolerance to the stresses. For example, drought stress has been shown to increase plant tolerance toward Pseudomonas syringae botrytis cinerea. Now, let us discuss the process by which drought influences pathogen infection. Plants under drought stress enhance ABA level, which triggers a signaling cascade in guard cells. This signaling results in stomatal closure and inhibits stomatal opening, which prevent entry of pathogens and decreases the infection capacity on plants. Target reduction in guard cell causes stomatal closure by continuous efflux of a potassium ion and anions from the guard cells, sucrose removal and etc. As you can see in the diagram, there are two types of anion channels that are R type and S type, activated by cytosolic calcium, which was triggered by ABA. After activation of anion channels, anions are released from the guard cells through this channel, causing depolarization. Due to change in the membrane potential, deactivates potassium ion in channels and activates potassium out channel, resulting in K plus efflux from the guard cells. Continuous efflux of potassium ion and anion channels lead to loss of guard cell turgor, resulting in stomata closure. At the time of stomatal opening, vacuum carries 90% volume of the guard cells, and during stomatal closing, 90% of ions are firstly transported to cytosol from vacuole. Cytosolic calcium elevation activates vascular potassium ion channels proposed to provide a pathway for calcium ion induced potassium ion release from the vacuoles. After reaching cytosol, potassium ion and anion influx from the guard cells, hence water efflux from the guard cell and cause stomata closing. Role of cuticular wax and stomata. Drought induce stomatal closure and increase the cuticular wax layer, which further reduces the pathogen infection and prevents the entry of foliar pathogens. Here in the diagram, you can see that plants are more susceptible to pathogens as compared to plant under drought stress. Under drought stress, cuticular wax formation increases, and this is hydrophobic in nature and devoid of moisture, which inhibits the multiplication of pathogens and reduces the pathogen number and makes plant tolerant to pathogen. Role of callose in pathogen infection. Plant cell senses the drought stress and increases the ABA hormone, which enhances the callose formation in the apoplast of plant leaf, and callose act as a physical barrier to pathogen and decreases pathogen number on plants. As we know that bacterial cells multiply in the apoplast of cell, but accumulation of callose makes bacterial cell unable to multiply in the leaf cell. Drought-induced molecular signaling cascade affects pathogen infection. As we know, plants under drought stress increases the ABA level. Through ABA signaling pathway, increases the expression of transcription factor, which induce up regulation of PR gene, resulting in PR protein, which decrease the pathogen infection and provide resistance to pathogens. Last one is conclusion. Plants under drought stress enhance tolerance against pathogen infection. Under drought stress conditions, plants close stomata for preventing water loss and reduce the chance of pathogen entry. Plants under drought stress enhances ABA hormone, which further increases the accumulation of callose, which act as a barrier against pathogen infection. Drought-induced molecular signaling cascades affect pathogen infection. Thank you.